Hello, everybody, and welcome to our presentation, Advanced Predictive Modeling in Cell Line Development. Let me quickly introduce my colleague and myself. I am Monika Zauner, and I'm an automation scientist. I will be presenting the first part of this presentation, and afterwards my colleague Ali Safari will be taking over. He is a data scientist, and we both are working at the Product Development Department of Sartorius Cell Line Development Center of Excellence, located in Ulm, Germany. Let me go quickly over the agenda. First, we will introduce the general CLD workflow of the 4-cell CHO platform. Then we describe the implementation of the cell selector and other automation opportunities in CLD. Afterwards, the implementation of the predictive data modeling tool for early identification of high producers is shown. And subsequently, we show our results, give a short summary, and then come to the questions and answers. So first, the 4-cell CHO platform, CLD services. Cell line development is an integral part of the biopharmaceutical production process. In contrast to chemically synthesized drug molecules, the biopharmaceuticals, such as vaccines, antibodies, and anti-drug conjugates, are manufactured from biological production systems like mammalian cell lines. Successful cell line development must complete the tedious process of identifying an optimal clone, as well as thorough characterization and scalability studies in a short time frame while meeting regulatory requirements. At Sartorius, we meet these needs with our 4-cell CHO platform. The first stage of this platform is the generation of a robust cell line based on our stable CHO DG44 host cell line and optimized expression vector. This part is provided by our facility in Ulm, where we go from DNA to RCB in 9 weeks. Here we generate the stable and ready-to-scale clones with predictable protein titers of up to 10 grams per liter. Following stages are the protein characterization, cell banking, and biosafety testing. Essential for the success over all these stages is our high-performing Smart CHO media system. Our media are chemically defined, ready-to-use products, which provide optimal performances with our CHO platform for all scales. Key features of our 4-cell CLD service are the speed. We go from DNA to fully characterized RCB in 9 weeks. Then the high producers we can generate. We have new selection technologies for early prediction of clone performance. Our titers, we can show product titers of up to 10 grams per liter. Our expertise with over 240 CLD projects and over 10 years of constant platform optimization, as well as our versatility. The platform is suitable for MABs, bi- and multi-specifics, Fc fusion, enzymes, hormones, DTEs, and biosimilars. And furthermore, we would like to mention our customer focus and flexibility to provide customization of offerings to meet our customers' specific needs. Now coming to the automation technologies, we focus on the cell selector for improved clone generation, and shorter CLD timelines. I want to give an overview of the Sartorius cell line development workflow and the automation opportunities during these steps. It all starts with our CHO DG44 host transfection, pool generation, and selection. Subsequently, single-cell cloning is performed, which is one of the most important steps in cell line development. The process to isolate and select individual monoclonal cells carries both the risk of making an incorrect selection and thereby discarding clones that would outperform others in the final scale, as well as the potential for poor outgrowth after the cloning step. Therefore, special focus must be placed on this step of the work process. Starting from several thousand clones, this number needs to be narrowed down, posing a time and work-intense challenge. However, it also presents the opportunity to leverage automation to make this process more efficient and reproducible. Single-cell cloning in our cell line development is performed with the Sartorius cell selector, which we will be talking about more in detail in this presentation, especially in regard to the automation we could achieve utilizing the Sartorius cell selector in combination with AI and machine learning modeling we perform to optimize and standardize the clone selection. The subsequent clone expansion and pre-selection consists of frequently recurring time-consuming and labor-intensive tasks, which is ideal for automation applications, including automatic liquid handling, automatic product titer measurement in our Sartorius octet systems, further cell line analysis in the Sartorius InQSight or IQ screener instruments, and finally, to evaluate and select the best clones for RCB generation, our Sartorius Amber 15 systems for automatic fed batch process runs of up to 48 cultures in parallel. Now we want to focus on the Sartorius cell selector for single-cell cloning. The whole system consists of several parts. First, the nanowell plate, comprising thousands of 4 nanoliter nanowells at the bottom of each of these 24 wells. Second, the device itself with the nanowell plate stage, the inverted microscope for bright field phase contrast and fluorescence imaging, and the precision robotic arm for cell isolation. Then the software for image analysis and high producer identification. And finally, the high precision glass capillary for clone picking. The workflow for single-cell cloning using the cell selector is shown here. Cells are seeded into the nanowell plate at a density of about 2000 cells per well, which is the cell density that results in the most nanowells with only one monoclone. Of course, there will also be nanowells without cells as well as nanowells that contain more than one cell after seeding. The cell selector screens for monoclonality on day 0. Typically, over 500 monoclones per well can be found. The following days, clone growth of the monoclones is assessed. On day 4, clones are transferred from the nanowell plate to the next plate format. We tested the generation of different product clones using FACS or our cell selector, In yellow, you see the outgrowth of clones after FACS single-cell sorting. In black, after the cell selector. We found drastically improved outgrowth rates, and even for difficult products, we found reliable clone generation using the cell selector. In a next step, we implemented a bead-based secretion assay during the single-cell cloning. The assay is based on beads, which are added to the nanowells at day 1 after single-cell cloning. The beads are coated with protein A. Produced and secreted antibodies from the monoclonal cells in the nanowell get captured and bind to the beads. Using a secondary fluorescently labeled antibody, the secreted product can be detected via fluorescence intensity for every single cell clone. So, by using the bead secretion assay, early clone selection based on antibody titer production can be achieved. Determining cell growth and fluorescence intensity, a specific cell productivity can be determined. So, potential high-producing clones can be selected and isolated from non-producers already 4 days after single-cell cloning. How the data gathered by the cell selector is utilized for AI and machine learning modeling will be described by my colleague, Ali Safari.
So, hello everyone. As it was mentioned by Monika, I'm Ali, working as a data scientist, and I will take over from here. Thank you, Monika, for the very nice introduction regarding our platform for cell line development. So for my part, I would focus on the predictive data modeling that we have implemented with the support of Sartorius cell selector data output for early identification of high producers. So initially, let's have a look at the overall general workflow and pipeline from data collection to data modeling. And the goal here was to use cell selector data to be able to predict clones and identify clones which have a better performance and considered as high producers in AMBER 15 evaluation run. So starting with the data collection, we use the output data of cell selector, which parameters could be categorized into 2 different categories, cell parameters and also fluorescence parameters. And as the next step, the raw data set for 5 different products were used to train our model. And as a pre-processing step, we do the data normalization before representing the data for the data modeling. And in the next slides, it would be mentioned, why such a step is needed. And as the last step, the data modeling would be started using different machine learning algorithms like, principal component analysis, classification type of algorithms, which was done in SIMCA. And also 2 more machine learning algorithms like artificial neural network classifier and random forest classifier. And at the end of this step, we integrate the whole, let's say, algorithm or a strategy into a one-button solution in SIMCA, which would be very easy to use. And then as an output, we would get the ranking of clones based on high-producing clone selection strategy. And this would be fed to the cell selector Sartorius device again for picking up the clones. And this would be a constant training and optimization of model with more data sets every time that you can retrain your model with new data to get more added values. So we can start with the data collection part. As I mentioned, we use the output of cell selector device as the input of our data model. And then here what we are showing in the left-hand side picture is the pictures and the images that are captured by cell selector during the process from day 0 to day 4, from like lay down of the cells, monoclonality, outgrowth, and fluorescence productivity as an example for one low producer and one high producer. And what would happen at this step is that these images later on, using the image, the software of the cell selector for image analysis, with that help would be translated then into parameters in general, into two different type of parameters: cell parameters like area, diameter, total detected area, and also some fluorescence parameters. And then these parameters are, let's say, the variables that are used for our data model as an input. So the next step, as it was mentioned in the general workflow, is the data pre-processing, and more specifically data normalization. And here I'm showing an example why this normalization matters. So in this case, we are working with 5 different products and protein antibodies, more specifically, that when you look at the measurement data and the pictures, let's say categorized into variables, and you put them in a principal component analysis, you see that for non-normalized data or like raw data, you see some clustering effects, simply showing that the measurements and also the type of variables, the order of the magnitude for the variables can be very different from product to product. And this clustering effect for your different products, as it was shown in the left-hand side picture, and different colors are representing different products, can have a negative effect on your learning process of your model in a way that can bias the learning process. So to overcome this data clustering, we use the normalization strategy here as a pre-processing step at the top of the raw data. And what was observed was very promising, which was shown in the right-hand side PCA model, that by very, very high extent, you can get rid of these clustering effects, and more normalized and standardized data are presented. And at the end, after this step, your data set would be a better representation, let's say, of your measurement to be used for data modeling. But before going through the modeling strategies itself, sorry, it's good to have a look at the general challenges that we are dealing here. So the big challenge here is that you want to provide a predictive tool, consists of model, which is able to predict the titer values and capture all biological complexities of different products. Considering that in a cell selector step, cells are in a totally different conditions in comparison to the, let's say, Amber 15 scale, where you want to have your prediction. So basically, you want to use the data from a cell selector in a very early stage to be able to predict the amount of your product, or let's say in Amber 15. So the challenges here are that, for example, medium is very different. So we in cell selector step, we are using cloning medium, but in Amber 15, we are using a production medium. So the nature of the process from medium wise is very different. Cell status is also different. In cell selector, cells are separated and are single, but in Amber 15, they are kind of fully recovered cells. The condition is also different. In cell selector level, we don't have shaking and steering, but we have a very more controlled process in Amber 15, like steering and gassing. And of course, the cultivation is also very different. And that's why if you initially start with the data modeling part using one specific product, as it was shown in the bottom picture, you see very good model with high R square and predictivity power. But if you combine all different, let's say, clones from different products together, your model would struggle a lot. And that's one of the big challenges here. So to be able to overcome some of these challenges, we came up with the classification algorithms. So in general, the classification algorithms are a kind of supervised learning techniques and are a subset of machine learning algorithms that are used to categorize data into predefined classes or groups. So these algorithms are designed to learn from a set of input data where the class labels are known, which is referred to as like the training data. So the goal of the classification algorithm is to accurately predict the class labels of a new unseen instance based on the patterns it has learned from the training data. In a simpler word, it means that if your model, or in general, your models are struggling to predict the exact value of the titer in number 15 based on your cell selector variables, which is challenging, then is that possible to potentially predict if a specific clone is a high producer or low producer? For example, if what we see in the right-hand side picture, for let's say to understand this concept a bit better, is that you see a model which is trying to, let's say, have a good prediction of the clones for different classes, like, for example, class 1 and class 2. You can call class 1 as like high producer, class 2 as low producers, which are highlighted with two different colors. So your model is a bit struggling to predict the exact value of the titers. You see, for example, the R square of around 0.6, which is not perfect. But then, for example, if you look at the two boxes, is that, okay, now the model is struggling and there's like a challenge for predicting the exact value of the titer, but still this can be good in, let's say, categorizing the clones as a prediction into different classes like high producer and low producer. So with that, we can go to the workflow of how the model setup, model selection, and machine learning algorithms implementation looks like in our workflow. So what I'm showing here is the general workflow of machine learning classification algorithms. So we start with a PCA, principal component analysis classification, using all the data that we get from cell selector, label them based on their AMBER 15, let's say titer or antibody production into class 1 and 0, class 1 high producers, class 0 low producers. And then we have, this would be basically our training data set. And then we would have like a test data set with some new instances. And then every time when we train the model, we test the model. And then we do some variable reduction or optimization. So we find better correlations or which variables are correlating better, let's say, with your titer in AMBER 15. And then this loop would be continued till, you know, you get a set of variables that are correlating the most and providing the better results based on the PCA classification. And that would be your selected variables, which would be transferred to the next step, where we add two more machine learning algorithms: artificial neural network classifier and random forest classifier, which from their nature, they are using very different learning techniques. So this is what we call the multimodal approach to add more confidence to our selection strategy. To see, for example, if we can find the clones that all three models are agreeing on to be a high producer. And then we do the same testing process. But what is, let's say here, was done as an extra step is what we call hyper parameter model optimization. What does it mean for this kind of machine learning algorithms? The way that, let's say, the model can affect the results could be very different based on the parameters that are given to the model. So hyperparameter optimization in machine learning involves selecting the best set of hyperparameters for a learning algorithm to maximize its performance on a given data set, for example. So hyperparameters are the configuration settings used to structure the learning process, such as learning rate or number of, for example, layers in a neural network or the number of trees in a random forest. So unlike the model parameters, which are learned from the data, hyperparameters are set prior to the training process and can greatly affect the model's ability to generalize from training data to unseen data. And considering that at the end of this step, you will have your optimized models, let's say optimized PCA, optimized random forest, and optimized artificial neural network. Each of these models separately would give their weight, as we call, for example, the sum of classifier variable. And at the end, your clones, each clones which you are using as like a test data set would get a score based on these different models. For example, score 3 means that all three models are agreeing on this clone to be a high producer. And then you get like a rank as 3, 2, 1, or 0. And at the end, this would be integrate this, let's say, sophisticated algorithms of like multimodal approach, which was implemented in Python considering hyperparameter optimization, would be integrated into Simca in a one-button click to get the clone ranking. But then there's also like a process of best model selection, what kind of thresholds you are applying, how to evaluate to come up with the, let's say, best possible solution. So how to classify the training data set, this is very important, and how to select the best model, as I mentioned. So at the end, we tested our trained model in two different data sets, two Amber runs that we had. And based on this variable selection threshold evaluation loop of, let's say, the process, we came up with the best strategy or the best models, which were providing 73% correctness rate based on the confusion matrix that we see here. Confusion matrices are categorizing and are used categorizing your models and are used as a kind of metrics to evaluate your classification algorithms. So 73% here basically means that if I have, for example, 34 clones which are a member of my class 1, for example, high producers, 25 out of these 34 are classified correctly as class 1, and 9 of them were classified wrongly as class 2. So this is how you read your confusion matrix. But yeah, this was all about the model, the strategy, how it was implemented. But now we can have a look at some results that how this selection strategy looked like in a real world experiment. So what we did was we used our new selection strategy in a couple of experiments and compared it with the more conventional way of selecting clones, which is basically a kind of random approach using some productivity assay data. And we benchmark it for some different products. And what was seen, for example, for product A, was that we saw more than 40% increases in the average of top 4 clones, which are showed here, which were highlighted in red. And for product 2, more than 60% increase in the average of top 4 clones, which is very nice and significant increase. So it's not only about, let's say, the top 4 clones, but you see that the likelihood of selecting better clones is also increasing if you have a look at the average line of the total, let's say, clones in that amber run. So this is the 2 products. I also have like another example for product 3 here, which is also interesting to have a look. When you look at product 3 and compare the selection, the new selection strategy based on the data model and the random one, you see that, for example, in the left-hand side picture, that there's like an outlier there, and the highest, let's say, producing clone was not selected with the model, but with the random strategy. But that's, for example, what we can call like a lucky hit, because what you want to, and that's like I'm insisting again, what you want to achieve with the data model is not only selecting the best clone, but also increasing the likelihood of selecting clones with higher fed batch titer. For example, if you look at the right-hand side picture and get rid of that one outlier, which is significantly different with the average of, let's say, titer of the clones with the random strategy selection, you see that the likelihood, sorry, of the selection is really increasing from a random strategy to the data model strategy, and also the top 3-4 clones titer is also increasing a lot. So we also did another study and analysis to be able to prove or check the model robustness when we use our multi-model approach. So the idea here was that we used one product, like the data for one product. And for that specific products, the clones which were selected had different scores, like, for example, 2, 3, 1. As I mentioned before, 3 meaning that all 3 models are agreeing on this clone to be a high producer. And for 2 and 3, when we have the score 2 or 3, we call it, let's say, high confidence. And when we have 1, we call it low confidence. And then when we compare the antibody titer in number 15, the prediction which was predicted by the model, we see a significant difference between the clones that were selected with a higher confidence than the lower confidence. For example, for this product, we see more than 50% increase in the average of top 4 clones with more than, let's say, with the score more than 2 or equal to 2 in comparison to the one which we call low confidence. And that's also a kind of robustness proof of the multimodal approach that we are using here. So at the end, the question is that, okay, now, like, by leveraging AI, machine learnings, you can get a much significant and more significant and better performance, or let's say early identification of the clones that you were aiming. But then the next question is about the automation, because with using machine learning and leveraging AI, you want to make your lives easier also in the lab. So the idea was how we can make this type of complex modeling kind of usable and easy to train for our scientists in the lab. So, as I also mentioned before, we integrated the development of the machine learning methods, which was done in Python, into Simca, and then which is like a sartorial software for multivariate data analysis. And then we could integrate all of them together as a like one button click, which provides you a data output with ranking of the clones, as you see, for example, in this picture here. So model A says 1, means that this clone is a high producer. Model 2 says that, okay, 0. Model 3 says 0, and then sum of classifier. And at the end, you would rank the clones based on this sum of classifier, and then would output it again to the input it again to the cell selector, and then the picking would be done. So basically, this was a conversion of a multimodal approach to an automated, easy-to-use platform in Simca, and fast and efficient training time we could do for our operators in the lab and adapt it into operational units. And also shown that, like, with Simca could be used for much more than PCA, PLS, or OPLS type of models with integrating Python scripts. So with this, I would move to this slide about the overall efficiency of using AI and machine learning in clone selection and cell line development. Integration-wise, as I mentioned, at the end, it was easy to adapt, optimize, train, and transfer to our scientists in the lab as non-data scientists. And the advantage of the new selection technology is that it leads to selection of high producers for the tested products that we had. And the model is also very efficient. It runs in seconds, since the data is available and provides the results. And from the development perspective, it's also a selection method which is based on AI and machine learning methods rather than random picking, and hence a more scientific approach considering all data available. So as the last slide for the summary, I would like to summarize our talk here and about, for example, our 4-cell CHO platform, which we could decrease the time from DNA to RCB in 9 weeks with titer of up to 10 gram per liter. So we have proved the process scalability and the clone stability. We implemented a new selection technology to find the high producer using cell selector data, using data analytics, AI, machine learning algorithms, and doing the automation in the lab. And then the new selection technology, as it was mentioned, based on machine learning algorithms, leads to selection of higher producers, significant differences in comparison to more conventional way of random picking for the tested products. And at the end, we provided a platform which is easy to adapt, optimize, and train and transfer to non-data scientists. So with this, I will finish the presentation here. Thank you for joining, and we are ready for the questions and answer, and would be happy to get your questions and feedbacks. Thank you.